In 1953, Watson and Crick, as a result of their model-building studies based on the X-ray diffraction work of Franklin and Wilkins, proposed the double helix structure for what is now called B-form DNA. This work earned them the Nobel Prize. In higher organisms, DNA forms a complex with histone proteins to form chromatin. Prior to the work of Watson and Crick, the scientific community had assumed that it was the protein in chromatin which contained the genetic information. Until the structure of DNA was modelled, no credible mechanism for molecular reproduction had been advanced.
The reason for the change in belief was that the Watson-Crick structure showed how directly how DNA might replicate in terms of two complementary strands. And when people change scientific ideas, it's not enough to have a hypothesis, which sounds plausible. You have to show there's a mechanism. And the DNA structure on its own showed that there might be such a simple mechanism. I think that's what convinced most people.
Perhaps the most astonishing thing about the original Watson-Crick discovery was the way in which the structure itself encompassed the biological function. And Watson and Crick in their great original paper, saw that clearly and discussed all of the major functions of DNA that were likely to be evident, which were evident just by looking at the structure. In some ways, you could say that many of us have been disappointed over the years that life is not altogether as simple as that original great discovery might have suggested it would be.
One remaining problem is that of how 2 meters of DNA can be packaged into the nucleus of every living cell, a nucleus only a few millionths of a meter in diameter.
The problem we have is that we have an enormous stretch of DNA that has to be compacted into a rather small nucleus. It has to be packaged. This might be perhaps not such a difficult problem if it weren't for the fact that the DNA also has to be ready to perform its biological functions. So you can't simply stuff it into your bag the way you would your dirty laundry at the end of a trip. It has to be packed neatly so that it can be unraveled when it's needed.
The human cell contains 46 or 48 chromosomes, and the DNA in one of them, if stretched out in a line, would be several centimeters long, and this has to be put into a space of a few microns. Now, the way that this is overcome is to package the DNA in a series of levels of organization, which we've come to understand in the last 10 years or so.
The packing problem can be dramatically illustrated like this. Here is an electron microscope picture of just one chromosome from a cell. If we break the cell, we see this. With the structure destroyed, the DNA spills out.
In this picture, one can still see the mitotic scaffold, the axis of the chromosome. The DNA is spilled out into this halo, and each of the little black lines is a piece of DNA double helix, and this is one long molecule. Now, DNA itself is a stiff molecule, and on its own it would run for past 500 or 600 angstroms without changing direction. So it is stiff, like a wire cable. What the histones do is to fold the DNA up into structural elements called nucleosomes. And this picture shows that there are histones which form a core around which the DNA coils.
So the first level of packing consists of wrapping 200 base pairs of the DNA molecule around protein core particles made up of histones. The histones form an octamer, a group of 8 molecules, and each octamer is joined by linker DNA.
At the lowest level of organization, we have, of course, the nucleosome, in which the DNA is wrapped in 2 superhelical turns about this histone octamer. But that's only the beginning. We have then an extended polynucleosome string, which has to be wound up further. There's some question still about the details of the packing, but most people believe that the way in which that's done is to take that string and to wind it into a superhelix with about 6 nucleosomes per turn, which looks, we think, something like this. Here are the individual nucleosome core particles with their 2 turns of DNA. We just see the DNA path here. And then each such particle is connected to its neighbor by the linker or spacer DNA. And then you just see the front 3 here, but there are 3 in back as well. So it's wound up in a solenoidal array with a diameter of about 30 nanometers or 300 angstrom. That diameter is not precisely, it's not uniform. It's clear when you look in the electron microscope at photographs from the electron microscope that there may be as many as 7 or as few as 5 nucleosomes per turn in some cases. That means that we're not going to be able to solve the problem of packaging at this level by crystallographic techniques directly. We have to use other less precise physical methods to find out what we do know.
The 30 nanometer fiber represents the next order of structural packing of the DNA, helping to explain how it can be contained within the nucleus. The coiled coil is not the end of the story, however.
It's still not the fully folded chromosome we reached. We've still got another 2 levels to go, and as we go up to higher levels, our information gets weaker and weaker. There's more of it, but it's more indirect. But the picture that I think is being convincingly built up is that the 300 angstrom fiber is folded into loops, the loops I showed in the original picture, certainly for the DNA which is inactive during cell division.
These so-called looped domains bring about a further stage of chromatin condensation. This level of organization is less regular than those previously discussed. Some regions of the DNA can be seen in the electron microscope to be more tightly coiled than others. The more open regions of the DNA are thought to represent areas of gene activity. So, to remain accessible, some portions of the DNA do not remain tightly coiled at all times.
We know even less about what happens next, but one model that seems most reasonable involves the attachment of this 30 nanometer fiber structure to a central nuclear scaffold, a central scaffold of protein from which 30 nanometer fibers loop out and then come back. In a typical loop, there may be of the order of 100,000 base pairs of DNA. And now you have this continuous stretch of DNA, of course, coming in loops like this in all directions to form a brush-like structure. And that brush-like structure then gives you the ultimate compaction that you need in order to fit the DNA into the nucleus.
So here's the complete story, from naked DNA to DNA packed into a cell. So much for the model, but how do we know so much? To get detailed information at the molecular level, chromatin structure has been investigated by X-ray diffraction. To do this, pure crystals have to be prepared from mixtures of DNA and histones. DNA and protein preparations are purified and then combined as a first step towards the growth over several days of the crystals to be studied. Next, small drops of the mixtures are placed on cover slips and inverted over wells, which maintain optimum conditions for crystal growth. Vapor exchange between the solution in the well and that in the drop encourages the formation of crystals. Crystals are composed of regular arrays of molecules so that the diffraction pattern corresponding to the molecular structure can be collected rapidly. Dr. Tim Richmond has been studying these crystals in great detail.
That particle was crystallized, and we could then study it by X-ray diffraction methods, which would allow us to actually look at the electron density of this particle. Now, the study is carried on to the level of 7 angstroms resolution. At 7 angstroms, we can't see each atom of the structure, but we can see the details, features, larger scale features of the structure, such as in the DNA, we can see the major and minor groove and the positions of the phosphate backbones.
Using an X-ray camera like this, X-rays are scattered by the electrons of the structure to produce a diffraction pattern. The crystal itself is mounted on the end of a small rigid fibre which permits reorientation of the crystal in the X-ray beam during the experiment. At present, there is no method of recombining the X-rays directly to form an image. However, the scattered X-rays form a pattern that can be studied. The position of the diffraction spots and their relative intensities can be used to generate an electron density map. Studying chromatin structure like this can give us an insight into its function.
There are two kinds of biological activities that DNA has to engage in a living cell. The first kind is that it has to duplicate itself. When a cell divides, you need to make a copy of the genetic material. That's called replication. Actually, we don't know a great deal about the mechanisms of replication in higher organisms. It's a difficult problem. A lot of research has been done, but the results are contradictory and confusing. The second function of DNA is to serve as a template for the production of messenger RNA, which is the working signal that codes for proteins and programs the cell for its particular functions. The process of copying the DNA into RNA is called transcription, and it involves an enzyme, RNA polymerase, that runs along the DNA, opening the duplex and using one of the two strands as a template for the production of a complementary RNA strand called the messenger RNA. Now, in order to do that, it's obvious if you just look even at this 30 nanometer fiber, it's not going to be too easy as a physical problem it's a physical problem for the polymerase to work its way through such a compact structure. So it's clear that this structure must in some way be disrupted in order for a gene to be expressed, that is, for it to be transcribed.
The problem of transcription can be compared to a film. All the information is contained within the film, just as the genetic code is contained within the DNA. However, the film is viewed at a distance by projecting it onto a screen. Similarly, the genetic code is manifested by the synthesis of protein molecules in a cell. But the cell requires different proteins for different functions at different times and for varying durations. So, just as there must be mechanisms for starting and stopping the film at the appropriate frame, transcription of the DNA must begin and end at the proper places in order that correct synthesis of proteins can take place. All these processes can occur in spite of the packaging of the DNA into chromatin.
Now, histones are regarded as a nuisance because they get in the way of things, but there they are. And it seems that in certain genes, the majority of genes, which are moderatively active, that is transcribing at a moderate rate, the histones are present and do not stray far away. As the polymerase, which is transcribing, passes, it can push them off, or they get displaced by some transient mechanism.
It is now thought that certain proteins, the polymerases, are responsible for the transcription of DNA. The ways in which proteins interact with DNA have been studied in detail. The interaction of X-rays with electrons in the protein structure allows the plotting of an electron density map. When proteins are studied at this level of detail, characteristic structures are found. One such structure is the alpha helix. It's a coil of amino acids. We can see clearly that there is a fit between one of the alpha helices in the protein and the major groove of the DNA double helix. The exact nature of recognition in molecular terms is still being investigated, but the basis for the recognition process is clear.
The protein contains 5 alpha helices within each monomer. And so the dimer of this protein has 2 alpha helices, one within each domain, that form essentially protruding ridges on the surface of the protein. And the structure of the protein is complementary to the structure of bDNA because these helices, helix number 3 within each domain, can fit directly into the major groove of B-form DNA. The fit is actually very comfortable steric arrangement, and in fact as early as 1959 there had been predictions that DNA binding proteins might use alpha helices to recognize B-form DNA. We found that the most reasonable models could be obtained that way, and it appeared that specific side chains on the surface of this helix made contacts within the major groove, contacting the edges of the base pairs, and thereby providing the specificity that allowed the repressor to recognize this one particular site out of the millions of sites possible within the E. coli chromosome.
The most detailed study so far of the way genes are turned on and off has been carried out by Professor Mark Potashny. He used a simple model system, the bacteriophage lambda.
It's been recognized for a long time that during development of an organism from a fertilized egg, genes are turned on and off in sequences so that different cells make different products and in fact form different parts of the body. And the question has been, how is this done? What's the molecular basis for turning of the mechanism by which genes are turned on and off? These genes must respond to external signals sent by other cells and by perhaps things they sense in the environment, as well as according to internal programs. In the 1940s, a group of Frenchmen at the Institut Pasteur, in particular Francois Jacob, Jacques Monod, Elie Volman, had the brilliant insight to recognize that these problems could be studied at the molecular level in bacteria. In bacteria we have examples of genes that are turned on and off, in some cases in response to extracellular signals. One of the paradigmatic cases of that kind of that phenomenon involved this virus, this phage called lambda. And as they showed, they and many co-workers showed, this virus has the remarkable ability to inject its DNA into a bacterium and then have that DNA turn itself off so that the bacterium can divide many times with the silent genes of the virus contained in it. We knew there was a molecule. We didn't know what the molecule was, and we had no assay for it. And so, to make a very long story very short, we had to devise special means for getting our hands on this molecule. But once we did, we were able to show that, in fact, it had the remarkable ability to look at the hundreds of thousands of sequences along DNA molecules and pick just the one that it was supposed to see and bind there and nowhere else.
In a schematic representation like this, the DNA appears completely uniform. However, the regulation of gene activity depends on a precise recognition of special sites on the DNA by specific proteins.
It's very unlikely that the details of the switch will be exactly the same in other cases. But there are certain fundamental principles involved that we think are very broadly used throughout nature. The fundamental principles being, how does the protein see the DNA molecule? How does a protein, once it's bound, turn a gene on or turn a gene off? So, for example, if we want to look more deeply now and see how does a protein see a DNA molecule, we now know from a variety of experiments, including structural work from X-ray crystallography, that that the answer is really quite simple. A simple representation of an alpha helix, a common structural motif of many proteins. The backbone of the helix is enclosed in a barrel, and protruding from the surface of the barrel are these side chains, which are characteristic of the particular amino acids that make up that alpha helix. Some of these side chains come off the front. Now you can see how neatly an alpha helix fits into a major groove of DNA. So we needn't worry that DNA has to assume a special shape or that the strands have to come apart. Instead, we recognize that essentially the protein has a structure which is complementary to the structure of the DNA, in the sense that it has a protruding alpha helix which fits neatly into the groove of the DNA. If we were to stick our nose into the major groove, we'd see the edges of the bases as they are found in the ladder. And the simple point is that there are simple chemical reactions, hydrogen bonds, for example, between these side chains of the alpha helix and functional groups that are exposed along the edges of the bases.
In some cases, proteins attached to the DNA act cooperatively. So, their precise relative positions on the DNA are critical to correct function.
One often has two sites for the binding of two different proteins. Both proteins bind, and then clearly the DNA bends in such a way that the proteins come into contact with one another. If you take the two binding sites and separate them somewhat more, you can study how the change in separation affects the biological activity. And in a number of cases, what is found is that as long as you put in about 10 base pairs of DNA, add DNA in multiples of 10 base pairs, everything is fine. 10 base pairs roughly is the repeat of the double helix. So you rotate the binding site through a full circle of 360 degrees. On the other hand, if you put in only 5 base pairs or 15 base pairs, then the binding site is rotated 180 degrees, and now protein 2 is facing in the opposite direction relative to protein 1. If you now bend, you don't make contact, and the biological activity is suppressed. So it's clear that DNA, that's the demonstration that DNA bending is important in biological function.
When Watson and Crick were living in this house in Cambridge, the science of molecular genetics was both young and seemingly well advanced. Their brilliant discovery was only the beginning, however. Much remained and still remains to be discovered.
Individual organisms have their own ways of solving many of the details of biological function, and therefore that you can't make generalizations without a great deal of caution. On the other hand, it does provide a great deal of pleasure and enjoyment for those of us who study the details.
Even those who believed the DNA was the genetic material from the experiments of Adrian MacLeod and indeed some other supporting experiments couldn't have appreciated that the structure of DNA itself would tell us how it worked.
There's no intellectual complexity to much of what we are discovering, except the complexity that nature puts in.